My PhD research and interest can be summarized with the following title: Characterization of proteins and protein complexes from commonly used biological buffers and physiological concentration of sodium ions by native mass spectrometry. But this really involves other metal ions at physiological levels, of course.
Hi, my name is Erick Baez. I got my bachelor from Universidad Central de Venezuela. Then I worked for several years in Procter & Gamble, where I realized I was interested in mass spectrometry. In 2018, I traveled to Canada to start a postgraduate program at the University of Alberta. Great experience. There I focused on method development for binding affinity measurements of protein-glycan interactions using native SIMS, or also called native mass spectrometry. In 2021, I had the privilege to be accepted by the Department of Chemistry at Purdue University, but it wasn't until September 2023 when I joined the McLaughlin group, and that's the research I will be highlighting today. Currently, I am a fifth-year PhD student. I will be graduating in April. Now, to understand the motivation behind my research work, I need to answer the following question. Why do we need non-volatile salts in native mass spectrometry when we know that non-volatile salts are commonly removed in native mass spectrometry due to metal ion adduction, which causes peak broadening and ultimately affects the mass accuracy of the measurement? It also introduces chemical noise and ionization suppression, which translates into loss of sensitivity. Unfortunately, sample desalting comes with a sacrifice of the initial amount of starting material and potential changes in protein conformation and dynamics. And I will bring up a very specific system to show you how important this is. This is the interaction of this enzyme with a carbohydrate. This enzyme plays an important role in our immune system. It is a carbohydrate binding protein, so it is a lectin, and it is considered a C-type leptin because interaction depends on the presence of calcium ions. Now, to perform this kind of binding affinity measurements, relatively high concentration of calcium ions are required, and the reason is that for the interaction to occur, the calcium ion binding sites on the protein must be fully occupied. The message here is that depending on the protein or protein complex, metal ions may be important for biophysical measurements and structural biology studies. This is why it is important to consider physiological concentration of the relevant metal ions depending on the protein system we are interested in, especially native mass spectrometry. That's where we identify a gap. We need native mass spectrometry-based method with increased tolerance for physiological concentration of non-volatile salts, and that has been the focus of my PhD work. To make a long story short, we published a couple of manuscripts last year where we basically demonstrated that the implementation of theta emitters is very useful for the mass analysis of proteins and protein complexes, dissolving physiologically relevant concentration of sodium and commonly used biological buffers. Although we didn't characterize extensively the mechanism behind the implementation of theta emitters, we reached the conclusion that implementation of theta emitters manipulates ESI droplet composition. This is a cartoon representation of a theta emitter. They are made out of the same glass material of a single barrel or a conventional nano SI emitter, but they have a septum in the middle that split the capillary into two channels. One of the channels contains the protein dissolved in physiologically relevant solution conditions plus commonly used biological buffers. The other channel contains ammonium acetate with or without solution additive. An important practical consideration is that the internal diameter of each of the channels of the theta emitter, this is the one that we use in our work, is approximately 1.4 micrometer. The internal diameter of a conventional nano-SI emitter generally goes from 1 to 2 micrometers. Once electrospray process starts, a Taylor comb is formed and rapid mixing occurs. But rapid mixing is a stochastic process. There will be several ESI droplet population formed during mixing. One of those ESI droplets population is depleted or relatively depleted of non-volatile salts, and those are the ESI droplets that will yield charged state resolved ions in the spectra, while most of the ESI droplets will have either high concentration of non-volatile salts or concentration resembling bulk solution. They will be responsible of the chemical noise you will observe in the spectra. We also use a couple of gas phase methods to aid in the dissociation of non-specific metal ion adduction while avoiding protein fragmentation or protein complex dissociation. This is representative spectra of a solution of beta-gal. Beta-gal is a tetramer of approximately 466 kilodalton, dissolved in 150 millimolar sodium chloride plus 25 millimolar HCl as a buffer system. As you can observe from the spectra, the implementation of 30 meters with 1 millimolar ammonium bromine solution additive offer a higher quality spectrum compared with the same method without solution additive. Overall, 30 meters with solution additives offers better signal-to-noise ratio, reproducibility, and repeatability. In this slide, I want to show you an application of the method implementing 30 meters with solution additive. I wanted to probe conformational changes of a protein. I chose alpha-synuclein because it is an intrinsically disordered protein and its conformational states are well known to depend on solution conditions. Also, alpha-synuclein is involved in Parkinson's disease development, so it is therapeutically relevant. What we found out is that alpha-synuclein dissolved in ammonium acetate adopts different conformations compared to physiologically relevant conditions. And importantly, the results obtained in the gas phase correlate with in-solution assays under similar conditions. These are representative spectra. At the top, you observe a spectrum of alpha-synuclein dissolved in 150 millimolar ammonium acetate, 25 millimolar tris, pH 7.4. These conditions resemble the extracellular environment. This is in one channel of the 30 meter. The other channel contains ammonium acetate plus 1 millimolar ammonium bromide as solution additive. The spectrum at the bottom is almost the same experiment, but this time ammonium acetate was totally replaced for sodium chloride. As you can observe from the charge state distribution of the spectrum at the bottom, there is a more heterogeneous ion population of the protein when the protein is dissolved in sodium chloride. And you can observe this better in the plot of relative abundances versus charge state obtained using 3 independent replicates. We also use conditions that resemble the cytosol and the lysosome. We submitted this manuscript we hope to see published soon. Conclusions. Manipulations of ESI droplet composition underlies the resulting effect observed with 30 meters. Solution additive delivered by 30 meters increased signal-to-noise ratio, reproducibility, and repeatability. And 30 meters can be helpful in protein-centric structural biology studies using native mass spectrometry. I want to acknowledge my group, my mentor, the NIH for funding, and the consortium for top-down proteomics for this opportunity. I have been following the activities of the consortium for quite some time, and it is an honor to somehow contribute with the consortium. Thanks.